Alright, the first two attempts failed to make this video, so I don't know what's wrong with my software. Let's try that one last time. Trying here to make a good review video for you for the male reproductive system. So you can hopefully study that during the weekend, and so you're not overwhelmed having to have a test on Thursday. As we agreed together, the primary sex organ we have, or the gonad in the male, is the testis. Now, we are not going to cover the gonadal differentiation or the sex differentiation between the male and female. We are just jump starting directly into the male reproductive system. Doesn't mean that the sexual differentiation is not important, but we're not just covering it in this particular video. Okay, so the primary sex organ is the testis. The testis, we agreed that it is in the scrotal sac, so it has a sub-temperature, which is important to maintain the normal functions of the testis. What are those normal functions? Production of sperms, production of steroids, mainly the testosterone, although it's not the only steroid. And so we'll see how that is regulated later on. The fact that the testis is surrounded with a sub-temperature or lower temperature than that of the body, and the importance of the cremasteric muscle as we covered in class. So if we look here, we'll see that the testes can be pulled up by the cremaster or cremasteric muscle, and therefore bringing it closer to the body, and that will warm up the testis. And so it provides you some kind of regulatory mechanism to maintain the temperature, not to be too cold, not to be too warm, depending on the relaxation and the contraction of the cremaster muscle. In addition, which is something you don't necessarily have to know from a physiological point of view, but in anatomy, we know that the pampiniform plexus, which are the venous plexus surrounding the spermatic cord, they're also important in cooling down the blood that is coming through the testicular artery, and therefore the blood that's coming will be also colder than the normal, than otherwise the 37 degrees. So there are more than one mechanism to maintain and make sure that the testes are at a lower temperature than that present in your body. So descent of the testis can be interrupted, and we can see that more frequent in preemies, premature babies, because the testis didn't have time to descend. So essentially, if you give it more time, usually they descend on their own without surgical intervention, but sometimes you will require some surgical intervention. And where is the testis? It depends where it's stopped, whether it's abdominal testis and inguinal testis and prescrotal, and finally we have that's the normal position. Now, we don't have to remember that, but we just need to appreciate the fact that the testis is going to descend from where it started, where the ovary is in the pelvis, and it's going to descend it down into the scrotal sac. And some reports have been suggesting that pesticides and endocrine disruptors out there can increase the risk of what we call abdominal testis or pelvic testis or cryptorchid testis. Okay, so if we decapsulate, if we remove the capsule, the tunica albicans, and we look deeper into, and we have here the tunica vaginalis, and we'll see that the testis is separated by septa into little lobules inside, and each of those lobules will contain the seminiferous tubules. Seminiferous tubules are also surrounded by what we call interstitial cells. You see interstitial cells here, interstitial. There's another name for the interstitial cells called Leydig cells, and there will be always blood vessels surrounding it, some capillaries surrounding that. That is very important. Leydig cells are very important in the production of testosterone, but we're going to focus now first on the seminiferous tubule. Seminiferous tubules are primarily made by the Sertoli cells and the spermatogenic cells. Spermatogenic cells, what are they? You're going to see in a little bit. They are the type A spermatogonial stem cell, and then we have type B also, which is not in this picture. And then we have this primary spermatocytes, secondary spermatocytes, round spermatids, elongated spermatids, and then the mature spermatozoa. Okay. So in addition to those two cell types in the seminiferous tubules, we also agreed in the lecture that there is also a third one, which is myocytes, very thin myocytes that are surrounding the seminiferous tubules, which are helpful in squeezing out the sperms as they are developed. And they also provide you something, if you remember, called the KIT ligand, which makes sure that these cells will remain, because the myocytes will be right here, and those cells will remain in the stem cell nature, and they will not differentiate. And so the male do not run out of stem cells, and that's an interesting fact, and especially when we hold a comparison with the female counterpart. And so what you see here is the fact also that the Sertoli cells are very tight to each other, so they produce to you something called blood-testis barrier. They are feeding the spermatogenic cells as they are maturing. They will help in flushing away those spermatids. Those spermatids are going to lose all that cytoplasm, which will be phagocytosed by the Sertoli cells, and they will give you some fluid that will flush them away. Also, it will help in the endocrine processes of the testis, and we're going to cover the functions of the Sertoli cells in the next couple of slides. So let's start first with the testosterone. We agreed that the testosterone is being made by those cells. We call them Leydig cells, because the gentleman who discovered them or characterized them, his name was Leydig, German scientist. And they are responsive to a stimulant coming from the LH, luteinizing hormone. But what does the testosterone do to a male? Well, there are functions before birth, during the sexual differentiation and the reproductive development. If you remember, we said that the external genitalia will look more towards male under the influence of androgen, under the influence of testosterone. In addition to that, testosterone is going to activate the Wolffian ducts, and therefore the Wolffians are going to give you rise to the seminal vesicles, to the prostate, and to the epididymis, to the vas deferens, the things that you need for the male reproductive tract. In the meanwhile, the testosterone is going to activate another hormone from the Sertoli cells called Mullerian inhibitory factor, or Mullerian inhibitory substance, or anti-Mullerian hormone, which will suppress the Mullerian ducts. And the Mullerian ducts, if you remember, these are the ones that will give the uterus, the vagina, and the fallopian tubes. And so testosterone is important for that as well. So all of these things are changes that took place before birth, right? And then after birth in early childhood, testosterone also helps with the male sex characteristics of the boy. Boys look different than females, physique, behavior, games, they play different games. So the hormones, the sex hormones play a very important role in that, in establishing the male phenotype. At puberty, then the testosterone will be responsible for the second sex characteristics, and then the increase in the genital size. Then after puberty, then the spermatogenic process is helped by the testosterone, in addition to the follicle stimulating hormone. It is important for the sex drive. The female sex drive was maintained by the DHEA, but the male is maintained by the testosterone. It increases the red blood cells, if you remember, so it increases also, it's an anabolic steroid, so it increases the muscle mass, the bone mass, and increases the red blood cells. If you remember, the hematocrit value was higher in male than in female. So there are plenty of functions for the testosterone, and testosterone, some of its functions also are indirect, that the testosterone have to be converted fast to estrogen by an enzyme called aromatase, and the estrogen is needed for the function of the blood vessels, the central nervous system. It is needed for the closure of the growth plate. Therefore, boys don't continue to grow taller and taller. After puberty, there will be tons of testosterone, and testosterone will be converted to estrogen, and estrogen will shut down the growth plate. And so, like I said, there are reproductive functions of testosterone, there are non-reproductive functions of testosterone. We do need to remember those, and there will be a summary slide for that. But first, the process of making testosterone, you don't have to remember all of this. We already dealt with before in the adrenal steroidogenesis. If you remember, the first step is the side chain cleavage of cholesterol into pregnenolone. Pregnenolone can be converted to progesterone. Both of them can be hydroxylated into 17-hydroxypregnenolone, 17-hydroxyprogesterone. 17-hydroxypregnenolone gives you DHEA, which can be converted to androstenedione. And by the way, 17-hydroxyprogesterone can also be converted to androstenedione. All of this was happening at the adrenals, but also here is happening at the Leydig cells. However, in the Leydig cells, you continue further. In the adrenals, you stopped here. And in the Leydig cells, you will continue the process into making testosterone, which gets converted outside the Leydig cells into DHT. It also gets converted mainly in the Sertoli cells into estradiol, but also in other tissues. And we all agreed that the estradiol is needed for the bone functions, for the central nervous system function, for the closure of the epiphyseal plates, for also the blood vessels, the tone of the blood vessels. We know that older men with testosterone insufficiency, they can suffer from something very similar to hot flashes that are experienced after menopause in female. So an interesting fact here is that testosterone is not the strongest androgen. There is even a stronger version of testosterone. It's called dihydrotestosterone, which is formed by a reduction process by an enzyme called 5-alpha reductase. This guy is very well needed for the prostate. So if you want to limit the growth of the prostate, you give 5-alpha reductase inhibitor, and that will shrink the prostate, which is a treatment usually or often is used for prostate hyperplasia and to also fight prostate cancer. So as I told you before, the process of making testosterone in the testis is in the Leydig cells, and it is under the influence of a hormone called luteinizing hormone, or LH, which is secreted by the anterior pituitary gland. It will bind to the receptor. Receptor will activate the G protein, and that will activate the conversion of pregnenolone all the way down to testosterone. Remember, estradiol conversion doesn't really take place in the Leydig cells. It takes place outside the Leydig cells. So somehow this is not very accurate about the estradiol conversion happening inside the Leydig. It doesn't. Okay. So testosterone is important for the function of the testis. It's needed for the spermatogenesis. One fun fact here is that the testosterone concentration is about 1,000-fold in the testis higher than that in the blood. And why is that? Because we have something that concentrates it in the testis. It's called androgen binding protein, which is secreted by the Sertoli cells, the sustenacular cells, and that maintains the concentration inside the testis to be very high, much higher than that found in the blood. Now, there are effects due to the conversion to estradiol, effects on the skin, the hair, the liver, the fat, also on the bone growth, the epiphyseal plate, if you remember. That is related mainly to estradiol. The active form of testosterone, the dihydrotestosterone, is important for the prostate, the penis growth, and the bone density. And so a summary for the testosterone, you will see that in the next slide, but here just to remind you of the 5-alpha reductase and the conversion between testosterone and dihydrotestosterone. You probably have heard that name before, Propecia, on commercials on TV for shrinking benign prostate hyperplasia. So like I said, there are effects of testosterone before birth, sex-specific after birth, reproductive related and non-reproductive actions, which are essentially anabolic effects. Some people assume that it has an effect on aggressive behavior. There is a debate on that. Also, the testosterone is important for just the male behavior in general. Too much testosterone can give rise to acne because the sebaceous glands have receptors for androgen, and they will increase the secretion based on the availability of androgens. Okay, so that was the first part of our testicular function, which was the steroidogenic part. But there's another part here, which is the spermatogenic part. So we are making steroids, testosterone mainly, and the testosterone was needed in part for making sperms. The other hormone that will be needed will be your follicle stimulating hormone, or FSH, as you will see. If we look at the mature spermatozoa, it will look like that. It will have the head, will have the midpiece or the neck, and then will have the tail. The tail is primarily microtubules. The microtubules are capable of sliding over each other by the effect of some motor proteins. Myosins are present here, and that will give you the whipping action, but you will need energy for that, and therefore we're stacking all the mitochondria right where it's needed for the energy to be developed. But please remember that the cytoplasm is almost all gone, and you have the nucleus. The nucleus is much smaller than a normal nucleus because it's very, very tightly packed, extremely tightly packed, hypermethylated. It has a centromere N, which is very good because once you start the fertilization process and the first division of the zygote, you already have the centromere, so it's great. You don't have to start from scratch. What's interesting for us later on is this little sac we call the acrosome. And the acrosome will be very important for the process of fertilization. Keep an eye on the acrosome because we're going to need that later on when we get to the penetration of the zona pellucida and the enzymes that are needed to break the zona pellucida up. Okay? Alright. So spermatogenesis will remind us a lot of the biology that we studied many years ago. And we agreed that back then we start with a diploid cell, and the diploid cell is 22 pairs of homologous chromosomes and then the sex chromosomes. So it is 44XY in this case because that is male, right? So that's a diploid cell, which is your spermatogonial stem cell. That will divide into type A and type B spermatogonium. Type A will continue to be a spermatogonial stem cell. Type B will start to differentiate because it's moving away from the basement membrane. Once that happens, that's a mitotic division. Once that happens, then the type B spermatogonial cell will also divide into giving you rise to primary spermatocyte. The primary spermatocyte also came by mitotic division. So up till here, we have diploid, full diploid cells. Now, from there, you will start to enter the haploid division. The haploid divisions, or the meiotic divisions, is characterized, if you remember, by first meiotic and second meiotic. In the first meiotic, you assort the chromosomes, but they're duplicated chromosomes. In the second meiotic division, you separate the sister chromatids from each other. So in the first meiotic division, which will produce to us secondary spermatocyte, remember this was first primary spermatocyte, the secondary spermatocytes are duplicated haploids. So they are 22X or 22Y, but they're duplicated chromosomes. So, but keep an eye here because you will see that the 21X and 22X and the 22Y are still connected to each other, and that's for a reason, because the genes that are necessary for the sperms to mature are present on the X chromosome, not on the Y chromosome. So the Y spermatocyte, secondary spermatocyte, is relying on the X spermatocyte for full maturation. And therefore there is this big gap between them that connects the two cytoplasms together, which will continue even in the second meiotic division to give you rise to spermatids. you will see that the round spermatids also maintain that gap between them. Like I said, because the Y spermatids are relying on the X spermatids for maturation. That is a true haploid. Here we separated already the sister chromatids from each other, producing to a single strand haploid cells, 22X and 22Y. And those will start to mature, will start to lose the cytoplasm, and they will give you rise to what we call the mature spermatozoa. Now, we did talk about the spermatozoa. Here we will appreciate more the structure of the seminiferous tubules and the fact that the Sertoli cells, another name for the Sertoli cells, are the sustenacular cells. They are nursing and providing nutrition to those growing cells that have left the basement membrane. Also, it is important in concentrating the testosterone in the testis, because you will want 1,000-fold more testosterone in the testis, and that will be provided by a protein that's being secreted by the Sertoli cells called androgen binding protein. They form tight junctions here between each other, and that prevents the immune cells from entering, prevents anything that is water soluble from entering into the seminiferous tubules. We call that blood-testis barrier. They're also important in flushing away the sperms, and in a process called spermiation, which is both by fluid flushing, by a fluid called seminiferous fluid, which is secreted by the Sertoli cells, but also by an action of a plasmin activation, which activates something we have studied before called plasminogen, which becomes plasmin, and that will chew off the protein connections here, the fibrin connection that hold the spermatids to the Sertoli cells. They have endocrine functions. One of the endocrine functions is the conversion of testosterone to estrogen. Another is the production of a hormone called inhibin, which is important for the downregulation of the FSH production from the GnRH. So there are plenty of functions, and we do need to remember the functions of those Sertoli cells, and that will be summarized here in this slide. So it produces the blood-testis barrier, nourishing the spermatids and the spermatocytes, all the spermatogenic cells. Phagocytic action, because remember the sperms have lost a lot of the cytoplasm, so something has to remove that cytoplasm. During embryogenesis, if you remember, I told you there is something called anti-Mullerian hormone or Mullerian inhibitory substance or factor that will break down the Mullerian ducts and prevent the formation of ovaries, not ovaries, but ovarian tubes or fallopian tubes, and the uterus and the vagina, and that is called anti-Mullerian hormone. Well, it is secreted by the Sertoli cells. Under the influence of the testosterone, you need the testosterone to get it, but the anti-Mullerian hormone is secreted by your Sertoli cells. Other functions, the fluid that is needed to flush away the sperms to the epididymis, androgen binding protein. If you remember, I told you you want to concentrate the testosterone in the testis, having 1,000-fold or so testosterone in the testis than in the plasma. Okay, so once again, the Sertoli cells are important for flushing away the sperms by seminiferous fluid. It secretes the androgen binding protein, which concentrates the androgen in the testis. It also has hormonal functions. It converts testosterone to estrogen. It gives you inhibin, which goes back and downregulates the production of FSH by the anterior pituitary. It secretes plasminogen activator, which activates plasminogen to a plasmin, and plasmin chews away the fibrin adhesions, and that will allow the spermatids to swim freely. So it helps in the spermiation process. The summary of the control of the endocrine, the reproductive endocrine hormones here is depicted in this illustration. We have the GnRH, gonadotropin releasing hormone, which is secreted by the hypothalamus. That will stimulate the luteinizing hormone and the follicle stimulating hormone from the anterior pituitary. LH has its target on the Leydig cells. The receptor is on the Leydig cells, and that will stimulate the production of testosterone. Testosterone is needed for the making sperms, but also testosterone will go back and inhibit the GnRH and will inhibit the LH. Now, the FSH will stimulate the Sertoli cells, and Sertoli cells will help in the spermatogenic process also, in addition to the testosterone. But in addition, the Sertoli cells are secreting to the inhibin, and the inhibin will go back and inhibits the FSH secretion. Now, GnRH is known to be important for the puberty, because without it you don't get LH and FSH. But what makes it secreted at puberty, we're not really sure. Some people think it is because of the fat signals. So the fat will give you leptin, and leptin will increase the GnRH. There are other peptides that have been demonstrated to play a role also, like MPY, neuropeptide Y, and gamma-aminobutyric acid. We know that melatonin suppresses the GnRH slightly, and that can be attempted for kids with too early puberty to slow it down by giving them melatonin. Also melatonin, because it's a hormone of the darkness, it increases in animals that have seasonal mating. So in winter, when the day is shorter, they will have less GnRH, and that will translate into less LH and less FSH because they have more melatonin. But once the day gets shorter in the spring, then they start the mating activity. So that's how we know also that melatonin have a role in inhibition of the GnRH on the gonadotropin. Okay. We made the sperms, we understood the spermatogenic process, and once the sperms leave the seminiferous tubules, this process is called spermiation, and the sperms will be held in the epididymis and the vas deferens, and they will undergo maturation, they will undergo capacitation, and they will start to acquire more movement at that point. Most of the fluid which was used to flush away the sperms will get reabsorbed there in the epididymis and in the vas. And you remember the Sertoli cells gave you seminiferous fluid to flush away the sperms. We don't need that fluid anymore, and so we will concentrate the sperms, and it's held there, and at ejaculation the vas deferens, which has a lot of smooth muscle fibers, will start to propel further the sperms in the process of ejaculation. As the sperms are ejaculating, they will mix with the secretion with the accessory glands during ejaculation. So the biggest of them is, or the bulkiest in fluid secretion, will be the seminal vesicles. We have the seminal vesicles on two of them, one on each side. It will produce to you the seminal fluid, which makes the biggest bulk of the semen. Also produces the prostaglandin, with plenty of prostaglandins, which is kind of weird because we thought initially that the prostaglandins are being produced by the prostate, but then it proved wrong. But still they keep the name prostaglandin even though they have nothing to do with prostate. They're actually made by the seminal vesicles instead. It produces fructose and pyruvic acid, which are used by the mitochondria of the sperms to give you the energy. And it secretes to you fibrinogen, and that brings us back to the blood coagulation and the blood clotting. Semen, as it's deposited in a semi-gelatinous mass, which gets digested later on. So there's a process of coagulation and decoagulation, which is very similar to the process that we saw in the blood, with the exception that here we don't stabilize the fibrin. The fibrin still is kind of loose fibrin. And so you get the fibrinogen from there, but the clotting proteins, you will get them from the prostate, and you will see that in the next slide when we get to the prostate. But the fibrinogen is coming from here. So the prostate, on the other hand, is giving you the clotting enzymes, and it's giving you also the enzymes that will need it to be chewing off that clot later on. Like I said, the semen, when it's deposited, it's semi-gelatinous, and within 20-30 minutes it will become fluid again, and that helps in keeping the semen close to the opening of the cervix of the uterus for optimal fertilization possibilities. Now, in addition to the clotting and declotting, it produces a lot of alkaline fluid. It's not as much of a fluid as the seminal vesicle, but that alkaline fluid is needed also for neutralization of the acidity of the vagina and the male urethra, because acidity is lethal to the sperms. One of the fibrinolytic, which is fibrin degrading enzymes that is being produced by the prostate, is a very unique protein that's only produced here in the prostate. It's called PSA, which is short for prostate specific antigen. Now, the prostate specific antigen is one of those declotting proteins, but it's also interesting because of the fact that it's not anywhere else other than here in the prostate. It can be used as an indication for the development and the progression of prostate cancer. So it's a good biomarker. It's not a perfect biomarker, but it's a good biomarker for following up on cancer. Other thing we will get into when we get to the fertilization, if you remember, the sperms have lost most of its proteins, most of the cytoplasm, and so with that it will lose also a lot of the surface proteins, the channels, the calcium channels, the receptors, but those can be provided by the prostate. The prostate fluid will have those little vesicles, and we call them prostosomes, that will fuse to the sperm cell membrane, and they can be useful later on in the process of capacitation and in the process of activation later on of the sperms. So keep that in mind, keep the prostosomes in mind, because when we get to the calcium channels and the receptors of the sperms, we're going to need that kind of information to be reminded of. Okay, so these are two accessory glands that we covered, the seminal vesicles and the prostate glands. There is a tiny gland left. It's called bulbourethral gland, which does not contribute much to the ejaculation volume, but it contributes to lubrication during sex. It is increased with the sexual excitement. It's a clear, thin mucinous fluid that can help in the lubrication, but it can also flush away the acidity of the urethra and helps in the neutralization of the acidity of the vagina. Like we said, the acidity is kind of lethal to the sperms. So that was the male reproduction physiology that we dealt with in class. I tried to summarize it here for you. Hopefully you will find this useful during your review, and I will see you on Tuesday. Have a great day.